Swayam Prabha. Digital India. Educated India.
So, see here I have given you examples of three different kinds of compounds here. So, octahedral, tetrahedral and square planar. This is in terms of energy, this is more preferred, but in terms of steric hindrance, in terms of the size, this is what is preferred. So, it is different and if the size of the ion is, if the size of the ion is too small, then also, for example, manganate. Manganese is in the +7 oxidation state, size is very small. Then also it will form, and also it depends on the ligand that is present. So they form tetrahedral. Permanganate, it's tetrahedral. But here, in the case of cobalt, it can be in the +2 oxidation state, or it can be +3 oxidation state. So what is shown here is a +3, and these are the kind of compounds Werner used. Werner is called the founder of inorganic chemistry. He's the one, all these kinds of things, probably 100 years ago we did not know anything. So Werner came up with all this without any of the modern tools that are there today. The spectroscopy, magnetism, many things. Without that, he came up with so metal can have this kind of geometries. Octahedral, tetrahedral and square planar, and he used this kind of compounds. So particularly he has used cobalt in the +3 oxidation state, because that is one of the most stable compounds that you can form with the cobalt 3. That is again explained by the crystal field theory. Okay, so it will form, cobalt 3 will form octahedral geometry, and also it will have the most stable crystal field theory, energy it will achieve. That is one of the reasons why cobalt is used, Werner has used. But you don't know what is the crystal field theory, so I will not be able to explain, but then maybe I have one or two slides to show you what is crystal field theory is. Okay, so you can see here nickel is, when the chloride is present it is tetrahedral, but when cyanide is present it is square planar. That's something you probably may be knowing already. So again it depends on the ligand. Cn minus can take the electron density away from the metal, because of the Cn minus is charged, and also molecular orbital picture, all that if you recall. So it will be able to transfer probably part of the electron density to the antibonding orbital, and it will not be as charged as the neutral compound, for example chloride, but in the case of Cn minus, square planar, but in the case of chloride it is a tetrahedral. So see the difference between the two ligands. One has a square planar geometry, other has a tetrahedral geometry. And even with the simple ligands, platinum forms a square planar geometry. It is again because of the crystal field theory, that going from 3D, 4D and 5D, stabilization energy keeps increasing. Number of electrons that are added also have increased. Stabilization also increases, and hence, and the size is bigger. So now the ligands are too far away, so that steric hindrance will not arise. Hence, they remain as a square planar, and square planar in terms of energy is preferred, but in terms of the steric hindrance they are not preferred. So when the size increases, now ligands size is bigger, and then that hitting each other, that problem will not arise, and hence square planar. Even with a simple ligand like chloride, if you take 4 chlorides here, then still square planar. And here is one with 2 chloride and 2 ammonia, which is cisplatin, and transplatin is very transplatin is an anti-cancer agent. So that's why that platinum complex is put here. So if you understand the chemistry of these transition metals, their structure, geometry, properties, why they are colored, why they are magnetic. Some are magnetic, paramagnetic, some are diamagnetic. So these things if you understand, then you will be able to manipulate and do the chemistry to your advantage. So see if you see here, cobalt is pink in color, nickel is green in color, copper is pale blue in color, whereas zinc compound, they all contain same ligand, but metal in different oxidation state, so they have different color, correct? So that is because of the number of NPTEL electrons, and also because of the where nickel is placed in the 3D, going from left to right, more electrons are added, and then energy is also will be different, and the number of electrons also matter. So cobalt 2 plus 3 NPTEL electrons, 2 NPTEL electrons and 1 NPTEL electron, and then 0 NPTEL electron. So number of electrons also determine, in spite of them having all nickel water 6 ligands, copper 6 ligands, cobalt 6 ligand. So they have the identical geometry, but metal is different, color is different, because of the number of unpaired electrons that are present in these compounds are different, and also the nature of metal is different, and hence their colors are also different. So transition metals are colored due to the presence of unpaired electrons, in spite of them having the same kind of groups around the metal. So 6 is very common. Metals containing 6 groups around the metal have a most stable geometry, which is what is called octahedral geometry, and that will have the most preferred energy, and hence the octahedral complexes are very common. For example, if you look at this manganese, metal is same, but oxidation states are different. So manganese in the +2 state, very pale color, but manganese in the +3 state, color is different. Manganese oxide is insoluble. Here you see is the manganese in the +C oxidation state, and here is the +7, which is permanent. That's something that you are familiar with. So depending on how many electrons are present and also oxidation states also determine the color. Okay. So I told you that they contain unpaired electrons. So they can be paramagnetic or it can be diamagnetic. So bulk metals can be used as magnets. So here that we are talking about is a simple metal compounds which are soluble in solvents or in the solid state. They can be used as paramagnetic or diamagnetic. So in the cobalt 2 plus 3 NPTEL electrons. So whether when you take the sample and put it in the center of a magnet, whether it gets attracted or reflected, that tells you whether it is paramagnetic or diamagnetic, right? So that means transition metals are, they have the one of the properties is they are paramagnetic or diamagnetic. Yeah, so this is the oxidation state of 3D metals is what is shown here. That what you see here, 2 is the common oxidation state. Orange color 2 refers to chromium in +2, +3. Now here these are all +2, iron 2, cobalt 2, nickel 2, copper 2 and zinc 2. Chromium 2 plus is not very stable. Chromium 3 plus is stable, but these are all the most stable ones. In the case of copper, +1 is also stable, and +1 state copper containing +1. In the +1 state there are stable compounds. Cuprous chloride, that is very stable, but it has to be kept away from the moisture and the oxygen, otherwise it will get oxidized. So you can see here some of them do have a higher oxidation state, 3, 4, 5, 6, 7, and the manganese is the example here, +7, whereas chromium can go to +6, or vanadium can go to +5, or titanium can go to +4, and scandium. So that the group number also, so these, some of them do follow the oxidation state corresponding to the group number. Okay. So, but going from this side, oxidation state, higher oxidation states are not possible. So this is where the Werner made use of cobalt in the +3 oxidation state and made various different kinds of compounds. and you can see here, if the ligand around the cobalt metal is 5 ammonia and 1 chloride. Instead of chloride, if you replace chloride with bromide, iodide, 3 different kinds of compounds. And you can have, 5 ammonia or nitrate or sulfate or carbonate. One of you were asking whether sulfate can be a ligand. Sulfate is a ligand here. So various different kinds of compounds is prepared and shown that they have different color and different properties. And for showing and developing such kind of a chemistry which nobody knew before, so he was given a Nobel Prize, Zurich, Switzerland. So he is the is called founder of inorganic chemistry. That I am sure some you would have read in the textbook. And whether the ligand is in the inner coordination sphere or outer coordination sphere can also be easily found out by adding silver salts, right? So whether how many chlorides can be removed from the complex here. If you are removing 3 chlorides, what it means is all the 3 chlorides are outside the coordination sphere. This is all something that you have studied, right? And that's how we did. So various different kinds of ligands are possible. I have listed some of them here: sulfate, sulfite, nitrite, carbonate, iodide, hydroxide, amido, cyanide, thiocyanide, nitro. So these are all various different kinds of simple inorganic molecules what is written here, but organic is one of them. I have given you as a ethylene diamine, but you can have EDTA, for example, is something similar to this. Here is the duster. So EDTA, for example, ethylene diamine tetra. So you need to put ammonia. So 3 ammonia, if you react that with the chlorostic acid, so 3 of them you will get 3 of them will be replaced, and this is what is EDTA. Depending on the pH, you can have minus or proton. So if pH is acidic, it will be proton will be present. If it is basic, then it will be in the anionic form. Sodium hydroxide is used, O minus Na plus. Potassium hydroxide is used, O minus K plus. So this is one of the EDTA is a multidentate ligand, and this is a bidentate ligand, and these are all monodentate ligands. So that means if it has a group has a capacity to give only one pair of electron, then it is a monodentate. If it has a capacity to give 2 pair of electrons from 2 different sites, then it is bidentate, and here it is a multidentate, because nitrogen also can give also 3 ethylene diamine. You have 2 of this here, and so another one, that is what is ethylene. So in this ethylene diamine, you acetate attach 2 of them here because 2 protons. Similarly, 2 acetate there, 2 protons, and that is what is EDTA, right? So EDTA has 2 nitrogens, and then you have 2 on this side and 2 on that side. So totally 6 donors. So it is called a hexadentate ligand. Okay. So yeah, here is an example where ethylene diamine bidentate or acetate, acetyl acetonate is what is this called, where 2 acetones are combined with each other to give a bidentate ligand. Or an oxalate, for example here oxalic acid, remove the proton, then it is oxalate. here is another derivative of this. So instead of two hydrogens, you replace one hydrogen by the same group here, and that is what this should be an NH2 here, and so this becomes a tridentate. One nitrogen, two nitrogen, and three nitrogen, and this becomes 1, 2, 3, 4. And this is what is EDTA that I have written here. This nitrogen has a lone pair of electrons, so 2 plus 1, 4, 5. No, 2, 3, 4, 5, and 6. So it's an excidentate ligand, right? So various different kinds of complexes can be prepared using various kinds of ligands. So I will
Sir,
linear, square planar, tetrahedral, octahedral, and all that I showed you. Linear, if the ligand size is very big, you will not be able to make probably whole ligand come together. Only two ligands will be there, and also depends on the nature of the metal. So I will just rush through, and then because we don't have much time to go to the other part of the title that I spoke to you,
sir,
showed you in the beginning. See, this is what is the bonding bonding in metal complexes. So that is where the crystal field theory comes into picture, where your VSCPR theory or all other theories will not be applicable. So I will not go into these. Is something that you probably will learn in your 12th standard. Yeah.
so a bidentate ligand would donate two lone pairs via one coordinate bond.
How it will show one? There is only one lone pair.
So how is a ligand called bidentate?
No, because there is a pair on this and there is a pair on this. So for two different nitrogen it will donate. Then only it's called bidentate. Yeah. So depending on, so I will not go into the details of different colors. That you have to understand the bonding theory. That probably will be able to do it in your 12th class. I just show you that how the metal ions in biology play an important role. So this is just a brief introduction to the nature of the metals, their structure, bonding, color, properties. To show you that, a few slides I showed you, but then I will switch to, I will not go into the details of this, which you will be learning in your 12th class. Okay. So all this will tell you colors here, different oxygen state, same ligand. One is +2 and +3, but different color. And here different ligand, but oxygen states are same. So 5 ammonia, 1 chloride, here only ammonia, but the chromium is the common metal. So oxygen state are same, but ligands, if you change 1 ammonia by chloride, colors are totally different. So basically that crystal field theory, if you understand, then you will be able to explain the reason for the change in the color, which I will not go into because anyway you will learn in your 12th class. Something that I am going to present later now, that some of you later decide to do biology, then you will have an opportunity to understand and know much more than what I am showing you there. But otherwise, I want to just show you that the chemistry is very interesting and very important. In every sphere, it will have an application. Okay. So I will have to close here because otherwise I will not be able to show you the other part of the lecture, which is not very big, which is short only. So metal ions in biology. Do you need a break? Okay. So this periodic table I showed you already before. But this slide, what it emphasizes is where and all the metal ions play important role in biology. So metal ions are present in metalloproteins and metal enzymes. Enzymes are required for various different kinds of functions every day. Hemoglobin is one of them, which is a protein that transports oxygen for metabolism, right? Because higher organisms are multicellular organisms. So that means your skin cell, your hair, or your teeth, or everything different. So these multicellular organisms independently function, and they have to do their job. If your skin cells are not doing properly, then you have problem with the skin, right? If your hair cell is not doing properly, or your eye is not doing working properly, you have problem with seeing. So that means each one of these cells independently function, and to function you need energy, right? And that energy is transported through the blood cells to different parts of the body, and metabolism everywhere happens. And to do that, you need oxygen. There is an oxidant to convert carbohydrate into carbon dioxide and energy, right? So oxygen, how it is transported to different part of the body through the blood, it is transported using a media is blood. In the blood you have hemoglobin. Hemoglobin contains iron, and that iron transport the oxygen. Something that you are familiar with is hemoglobin, that's why I keep emphasizing on that. Okay, otherwise various different kinds of. So you can see here all the so many different kinds of proteins and enzymes. So about 1/3 of them that are known today contain metals. All animals, plants need metals, and they are very crucial for many daily biological functions, right? But also metals are used from outside as a diagnostic agent. That is one of them I showed you in, for example, magnetic resonance imaging. Gadolinium is introduced from outside, or cisplatin is used as an anti-cancer agent, one of the anti-cancer agents. So these are all introduced from outside, but many of them are already present in nature. So hemoglobin is one of them. Chlorophyll, if you take, magnesium is present, right? So metals in biology, they are present at the active site of the what I am showing you here is a protein.
Sir.
And that protein contains this backbone called a protoporphyrin. Pro, that means the first one to come to life. Yeah.
So in the previous slide, it was mentioned that it's also used for therapeutic applications.
Previous slide.
Along with diagnostic probes, what are the therapeutic applications of?
Therapy is treatment. The cisplatin, anti-cancer agent. That is what is therapy, na? Diagnostic probe means gadolinium is used to get an image of your brain wherever if it is affected. So it is used as a probe, but a cisplatin is used for treatment. So that is what is a therapy. So these are all the molecules that chlorophyll, vitamin B12, or the Popeye eats the green leaf. What is the green leaf that he eats? Popeye. None of you have watched the cartoon? Huh? Spinach. So spinach contains the iron sulfur cluster. So that means it is rich in iron. So since it is rich in iron, iron is necessary, Otherwise your blood is iron deficient, that means you are anemic, right? It will not be able to do the same function efficiently. So hence sometimes iron supplement is given. If you are very smart, then you would eat spinach because it is very rich in iron. All greens are rich in iron, but this is one of the richest. And to popularize that information, those cartoons were made. Popeye, that he keeps tin of spinach he eats, and you get so much of energy. So to popularize that, those cartoons were made. So they contain this iron sulfur cluster, and these iron sulfur clusters are present in our body. So hemoglobin.
Sir, in plant respiration, these FES clusters are present throughout the pathway, right? Respiratory pathway. In plant respiration, throughout the respiratory pathway, these are present in different places.
Correct.
So, what function do they perform? Like, are they just catalysts, or do they have anything more specific?
Now, the reason is that if you want to do a particular reduction of oxygen, if you want to do, it requires very high potential. So, in one step, if you want to do, that will damage the cell. So, it is done in different stages. And at different stages, these are the different kinds of proteins that are present, metal clusters are present. So, each one of them will transfer only one electron. At a time, 4 electrons, 6 electrons is transferred, that means redox potential is very, very high. And that is not good for cell. So, such a drastic change in the redox potential would damage the cell. So, that's why these are interspaced with one electron transfer. So, many different that's why it's called electron transport chain. So, at different stages, different proteins are present, and each one of them have the capacity to transfer electron from one site to another site. And the energy required also will be different, hence they are different proteins. It is not same one that is doing one electron transfer everywhere.
Yes, sir, but these iron sulfur clusters are present in the proteins, right? So, they are basically just catalysts, or do they have any specific function?
Electron transfer. They are electron transfer also.
They also transfer electrons.
They will transfer. These are also electron transfer proteins. In fact, this is electron transfer protein, but also comes from the spinach. So, if your body needs iron, there is a source. These are electron transfer. See, there are 4 iron. So, each one of them will have +2, +3 oxygen state. Change in the oxygen state will be there, and each time they can transfer only one electron. Higher electrons transfer requires higher potential, which is not good for the cell. So, it will damage the cell. So, that's why they are interspaced between many, many different kinds of proteins. Different proteins, because they have a different capacity. At some stage, a different potential, another protein will transfer that electron to another one. Not the same protein.
Thank you, sir.